All right.
Hey guys, Steve here, Ponics. Today we're going to talk about
Growing with fishes. Growing with fishes.
Hey everybody, welcome to Growing with Fishes podcast, episode 200 and or sorry, 352. A little bit off this evening. Hope you guys are all doing all right. Looks like we're having a couple of glitches. Oh, there we go. Now, how's everybody doing tonight? I hope you guys are doing good. Today we have Anna Schwabe back with us again. She's a really, really awesome person. We've had her on the show before, and I really appreciate you taking the time to join us again this week, Dr. Anna.
Thanks for having me. I love being on the show. I'm excited to talk to you again.
Um, before we get started, I just want to do a quick um, notes here, or housekeeping, I guess, for lack of a better term. I do have an aquaponic masterclass coming up May 22nd through 26th out in Beggs, Oklahoma. This is a 5-day course. This is the first time we've taught a full 5-day course. I've taught a 4-day version of this, but now we have so much information, especially on the microbial side. We really do need 5 days. So we'll be teaching this May 22nd through 26th in Beggs, Oklahoma. They have a really awesome facility out there, lots of different types of setups that we're going to go over in person. So you get, hands-on training, see what the different types of setups look like in person. And then also, get a chance to learn all different types of stuff. There's over 1,000 slides of education that we're going to go over, and lunch is included. We'll have all kinds of fun goodies and things like that for you guys to try out. So check that out over at aquaponicmasterclass.com. And we'll also be releasing this as a video format for online as well in June. So if you can't make it in person, just wait an extra couple weeks and we'll have that up online as well. So you should check that out if you're looking for all-encompassing aquaponics. It does touch on cannabis a little bit, but it's mainly geared towards all types of crops. We cover trees and root crops and a bunch of stuff that we don't cover in other types of courses that I've taught in the past, or haven't taught in a long time. We used to teach it in the advanced aquaponics class a long, long time ago, but never from top to bottom. So check it out along with Olive Branch Aquaponics. We'll be teaching it jointly with their farm. It'll be a great time. Alrighty. Thanks a lot for joining us again this week. You want to tell everybody a little bit about yourself and before we get started with the interview, and then we'll go from there. You're all kinds of awesome stuff. I actually saw you used to do work too at the Denver Botanical Garden, which I thought was super great. I used to go down there quite often when I lived in back in Colorado.
Yeah, so I can go ahead and give you hopefully what is a short but detailed little synopsis of my background. So originally from Australia, I moved to the States in 1987. So I've been here for quite a while, but I'm still an Australian citizen. We moved to Denver and I graduated from Cherry Creek High School. And then when I went to college, I went to CSU, Colorado State University, and I thought that maybe one day I started out I really wanted to be a marine biologist. And I thought that maybe like fish and wildlife would be similar. Turns out it's completely not similar. So I ended up changing my major to anatomy and physiology, thinking I might want to be a vet someday. And I went to CSU for 5 years. I didn't graduate my last semester there. I turned 21 and I didn't get good I didn't get any grade. I got 0 grades. I got Fs. I got straight Fs across the board. And then I took some time off and I worked in pizza. And then in about 2007, I decided I didn't want to work. I loved my job. I loved my team. I was the general manager, but I just didn't, there was nowhere for me to go. It wasn't challenging anymore. My store ran with or without me. And we were, just doing really well, and I wanted to do something else. So I decided to go back and finish my degree. So I transferred all my credits to the University of Northern Colorado, finished my bachelor's in cellular and molecular biology. And then while I was doing that, I ended up I ended up working in I needed to get like some lab skills, and you have to do that at UNC to graduate. You either have to do like a senior project or you have to work in a lab. Like you can't just graduate and not have any skills. So I worked in the plant population genetics lab, and I was, doing DNA extractions and working on this cactus project. It was pretty cool, but I wasn't really a plant person. Um, but I did have to take a plant class in order to graduate on time. Otherwise, I would have had to be there a whole nother year. I really wanted to take animal behavior, but the class was like over full. So I really had to take this plant class and I didn't want to take it. It was plant taxonomy, which is, the naming of like species and all that stuff. And looking at plant structures and flowers and leaf shape and blah, 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 boring. And anyway, we had to go and collect plants, and we had it was a brand new teacher. It was his first semester teaching, but he was like young and really excited about plants for some reason. I don't know. And he had a list of 100 must-know plants in Colorado, but we had to go out and collect plants. And the assignment was to collect 20 plants that are not on the must-know list. And I was like, that's like 100 plants. That's like all the plants in Colorado. There's nothing left to collect. I was so mad. I did not know anything about plants, clearly. So anyway, I did collect the plants, and part of the assignment for that was that we had to press them like a herbarium specimen, and we had to draw them like a little sketch to show that we understood like the leaf shape and the structure of the plant, and then we had to identify them and do all that. Turns out my teacher thought that I was a pretty good drawer, and he's like, "Oh, you should check out the Denver Botanic Gardens. They have a botanical illustration course." It turns out to be a whole nother like degree, which I ended up doing. So I have a certification in botanical illustration now. But anyway, that guy was Mitt McLaughlin, who was my mentor for my master's and my PhD. That lab that I was working in doing cactus stuff, I ended up taking that on as my master's. My master's project, which was looking at the hybridization levels of a rare cactus and introgression from a common cactus, and they're really, really closely related. And it was a land management situation. So this Sclerocactus glaucus, the rare one, grows in an area where they're doing a lot of gas and oil exploration. And so where the rare plants grow, they can't dig, right? Because they're protected species. And so what they wanted to know was if any of the populations are being introgressed by this common species, so that then they can not bother with those populations and dig there. And what we found was that really they're not hybridizing. And so the glaucus, the rare one, has hookless spines. Its common name is the Colorado hookless cactus. And the parviflorus has these hooked spines, and they were seeing populations with these hooked spines that were supposed to be glaucus, and they were like, oh, well, they're all hybridizing, and so we can just do whatever. Well, it turns out that morphological features are not great identifiers for species delineations, and genetic tools are much better to try and figure out who's more closely related and who isn't. And this relates to cannabis because, well, it relates to all plants. They express different phenotypes based on where they grow. And sometimes hooks are better, sometimes they're not. But anyway, it turns out that they're not really hybridizing that much. And within that rare, so the blue dots and the green dots are the rare species, and the red dots are populations of the common species. So we were looking at how much red is getting into the blue and green. We actually didn't know that there was a north population and a south population of glaucus, and they have split those, and they're managing them separately now as the north and the south group, because they are really different to the point where they could even be probably considered subspecies or even separate species, which was really interesting. So anyway, so that was my master's project. And then following that, I went to and this was in collaboration with the Botanic Gardens. And so after I finished my master's, I went and worked with the Botanic Gardens conservation department and ran their genetics lab. We worked on about 15 different projects, and that was about a 2-year job, and it was only supposed to be for 2 years. But while I was there, I had a volunteer who was really into cannabis, really into, he was actually getting into the industry, making edibles and vape pens and things like that. And we would have discussions. And it was about 2015 when Colorado legalized. And my job was ending. And I was like, I think I should go back to school and try to answer some of these questions surrounding cannabis. Like the first question that we thought about was, how come when you buy Blue Dream at 2 different dispensaries, they have completely different effects? They're completely different. They shouldn't be. And at least with my very little knowledge about cannabis, I was like, but they grow them from clones. Why are they completely different? Let's look at the genetics, because the genetics will show us, are they identical or are they different? So, that's a really easy question to ask and answer. So that's what I did. And so the school, the University of Northern Colorado, a lot of universities, and this is still the case, won't touch THC type cannabis at all because it's still federally illegal, and allowing cannabis research can potentially, what do I want to say? Put federal research, including FAFSA and things like that, in jeopardy. So they won't touch it. It just so happens that UNC is a small school. The, Mitt McLaughlin knows the dean, knows the provost, knows the president, and he went and talked to each one of them personally and discussed the project. And they all decided, yeah, that's okay as long as she doesn't bring any smokable cannabis onto campus. We can do that. And so I figured out a way to do like pre-extractions off campus and then bring the solutions onto campus and finish the DNA extractions. So I have this PhD in cannabis genetics, and I did several different projects. That was the first one. And what we found was, so there's 30 different strains from, I don't even remember how many dispensaries, 30 strains from, I don't know, it's in there somewhere. But yeah, 30 different strains, 122 samples. And what we found was that out of the 30 strains, we had multiple, obviously, samples within that strain to compare, are they the same or are they different? 27 of them had at least one outlier that wasn't a genetic match. So that being said, that answers the question when you go to different dispensaries and buy something with a strain name, how come it's different when it shouldn't be? But that's because they're not the same. And then, so that was really interesting. And people ask me all the time, like, okay, so what do I do then? Like, what do I do when I find something that I like? I'm like, go back to that dispensary and buy all that you can and put it in your freezer, because you may not ever find that exact thing again. Like maybe what you got was the anomalous version, and you might never ever find it again. So, which is really annoying, and I don't really have like a good fix for this. But you can see, like, if you go back up to that blue and yellow thing, so those are arranged by strain, and it goes from sativa up there in the top left is Durban poison. So it says 100. That just means it's been described as 100% sativa. And then down at the very bottom on the right is purple kush. That's 0% sativa because it's 100% indica. At least that's how it's described. And that's based on a combination of genetic lineage and effects and things that don't really go together. But that's how they're described online. That's how they're described in the dispensaries. So that's how I have arranged them. And so, like you can see in the Durban poisons, I think there's 8 or 9 of them. All of them have that strong blue genetic signal, but there's one that is clearly different. Like you're just looking, which of these things is not like the other, right? You don't have to know anything about this other than you're looking for the exact same thing on all the samples. One line equals one sample, one individual. So that Denver one whatever that Denver 1 dispensary is. And I didn't want to call out any particular dispensaries. So that Denver 1, whatever they're selling is different than what everybody else is selling. And there were, and that's the same for all the strains, except for a couple, like Jack Flash, which is on the bottom line, kind of a little bit in the middle, but to the, next one over, next to Girl Scout cookies, Jack Flash. So there's only 2 samples of those, and they both look pretty similar, but that's only 2 samples. I bet if we had 10, there would be definitely an outlier. So, so we found that really interesting. And one of the most interesting things I think about this study was the chem dog. So when I was going around to different dispensaries, I didn't know anything about chem dog, but I saw chem dog D, chem dog 1, chem dog DOG, chem dog DAWG, and I was like, what's up with all the chem dogs? Chem dog 4. But whenever I saw chem dog, I bought it, right? Because I figured either that maybe they're like, that dispensary's take on a chem dog, or maybe it's a next generation thing. For anyone who knows the legend of chem dog, it's about, this guy bought a pound of weed and he found 13 seeds, and he grew 4 seeds, which he called, I think it was, I don't know if it was 1, 2, 3, 4 or A, B, C, D, but that's where the D and the 4 and the 1 come from. It was Chem Dog 91 now. But there was only like one of those that he kept, a couple of them were males. One of them he didn't like. So he just kept 1, and that was Chem Dog, I think 1, and now it's Chem Dog 91. But then a few years later, he grew out 4 more and he called them A, B, C, and D. And so that's where the Chem Dog D comes from. And so if you look at the Chem Dogs, they are all a little bit different, but they're all pretty similar. And this is the kind of like relationship we would expect to see from seeds, all from, the same kind of grow. Like they're all a little bit different, like siblings are, but they're all still really closely related. So I thought that was like one of the coolest things about that particular study. And then the next thing I did was with an undergrad, and that was the one that included the University of Mississippi stuff, the research grade marijuana that everybody has been questioning for a while now. It doesn't smell like anything. It's got no terpenes in it. It's got a lot of CBN indicating that it's old. And it's got sticks and stems and leaves, and it's just not good, right? So this one's Daniella's. Oh, well, so when I got my hands on this Mississippi stuff, it was actually our chemistry department upstairs. They had a DEA license and were looking at cannabinoids, and they got their study material from the University of Mississippi. So I had access to it. And the funny thing is that when you have a DEA license to do research on research grade marijuana from NIDA, from the University of Mississippi, every single crumb of that material that they send you has to be present and accounted for. You can't, like, if anything's missing, they will do like an audit. You'll lose your license, probably get kicked out of school, whatever. So I was like, well, I don't need to take the plant material. I just need to take the DNA out of it, and then I can go back to my lab. And so Richard Hyslop, who's also on this paper, it was his lab. He was like, yeah, can you extract it in my lab and leave the plant material? And I was like, yeah. So he read through the licensing agreement and all this, and he's like, I don't see any reason why you can't. It says you can't take plant material from the lab. And, they keep it, that he had to keep it in storage, and the storage conditions were in a safe, locked, in a fridge that is bolted to both the floor and the wall, concrete, and that fridge also has to be locked, and everything in and out has to be like written down, weighed, all that stuff. So of course, we adhered to those rules, and I took my little tubies in and I walked out with my DNA. And so I extracted enough for myself and for Daniela, and I gave Daniela DNA for her study as well, because we wanted to compare because she uses different tools than I do. She uses next gen type sequencing, like the fancy expensive kind of genomic work, and I use cheap tools because I didn't have any funding for any of my work. And we wanted to see, like, do we get the same results? Where does NIDA happen to fall? And so if you look at this, you can see on the bottom, we've got wild hemp, we've got aruduralis in there, Carmangola, Colorado Gold, and those are all hemp, right? So hemp has this really strong signal that's in yellow, showing that they're all very closely genetically related. They have the same sort of signal, and they're all associated. And then when you start getting into the high CBD sativa hybrid indica, you've got these other genetic signals coming through that are indicated in purple and green. But those NIDA samples, which are right next to the hemp, they both belong with the hemp group. And we're not sure why that is, because it's not indicative of anything that, any patient is taking for any kind of medical, use. And a lot of the papers that have, and there aren't a lot of, studies using smokable marijuana, looking at any kind of indications, but anything that is out there, does smoking marijuana help with epilepsy? Studies say no. Well, if they're getting, and this is the only place you used to be able to get research grade marijuana, you couldn't get it from anywhere else. Well, yeah, if you're using this, of course it's not going to help, because it's not, it doesn't have the same cannabinoids in it. It doesn't even, it's not even genetically related to what people are purchasing at dispensaries and things like that. So, in this diagram here, we've got the hemp is in yellow and orange, and NIDA is those blue dots. And the non-hemp types, marijuana types, are the purple, red, and green. And those three purple squares, those are the high CBD types that are like CBD dominant, but over the threshold to be able to be called hemp. But yeah, so that one was really hard to get published because I mean, that's calling the feds out for giving people shit weed to do medical research on, right? And so finally we did get that published, but it was a long time to get that one published. That people were, the publishers were very wary about what the implications of this research are. But Daniela came out with the same paper, and I think we published in a special edition of this journal called Behind Smoke and Mirrors, and they were specifically looking for this type of research. And since Daniela and I both had, the same samples and showed the same thing, that NIDA is providing something completely different than what people are actually taking for medicine, we both got them published. So in her stuff, they're in the little green dots, and they don't really fall within the places you think that they should.
That's wild that it's basically wild hemp that they've been passing off as medicine. No wonder that they're not getting the same results as the more modern chemotypes.
Yeah, and I had a sample that was wild collected from I want to say it was 1969. And that was the closest sample genetically to what I had. And I'm thinking that, they started that program back in the late 50s, 60s. I'm like, are they still growing the same cannabis that they were growing back then? Have they just been saving seed and regrowing? Because cannabis has come a long way. Like, the wild growing hemp might not have been hemp. It might have been something that a hippie, a seed that a hippie flipped out the window. Like, who knows, right? But, or it could have been that they're growing outside for this stuff. So maybe it's been open pollinated and it's been intergressed with genetics from back in the day. We don't know, because they won't talk about it.
Yeah, that Mississippi pollinate this grow.
Well,
I
and
did want to go ahead.
the stuff that they do their research on, they grow indoors, like in pristine conditions. They take really good care of it. It's the medical, the research grade marijuana. They grow it outside and then they harvest it, they grind it up, and they store it in like these cardboard bins with no like they're not airtight, they're not, just like not temperature controlled, whatever. So like no wonder it is what it is.
I wanted to just ask you too. So you did work with high altitude cactus. I had an old roommate who used to sequence some of the high altitude cactuses in Colorado, and he was always saying how the DNA breaks down extremely quickly. I just wanted to ask if the species that you were working with had that same issue, where you had a kind of a shorter window in terms of sequencing them before the DNA got harder to sequence.
No. So what cactus are pretty slimy, right? And when you so these cactus, you can't really, it's not like a plant where you can take a leaf. Um, so because the leaf are the spines, and then the stem is the barrel. So all you can take is a flower. And so that's what was collected for the sampling. If the cactus only had one flower on it, then we couldn't sample. So so you put the flower into a little, paper bag or plastic bag, and then you add silica gel, which just pulls all the moisture out of it. So you've got a dried flower by the time it gets to me. And then we, pulverize it into a powder and then add the detergents and salts and all that stuff that end up taking the DNA out. But no, the DNA, we've still got DNA in freezers. I mean, you wouldn't want to leave any DNA out at room temperature for a while, but DNA is really stable. Um, if it wasn't, we'd be in trouble because you can't just have DNA falling apart when it gets warm. Um, I don't know why he would have problems with, unless he was doing RNA. RNA is not stable. It does degrade really, really quickly. You have to be pretty quick when you're working with RNA. That's the only thing I can think of.
Yeah, I just remember him talking about specifically with some of the cactuses that they were sequencing. There was some kind of problem. Again, this is like secondhand. I was just kind of curious
Yeah, yeah.
about the experience with your stuff.
Yeah, and
Um,
the thing with cactus is because they're slimy, they have a lot of secondary compounds that make them slimy, so it does make extracting the DNA challenging. I had to kind of create a new protocol to do that, which came in very handy with working with cannabis because cannabis also has a lot of secondary compounds that can make it very difficult to extract and clean, right? So,
Interesting. Um, you had also done an human olfactory discrimination genetic variation paper, and I was hoping you could tell us a bit about that because we haven't
yeah.
had a whole lot of people speak on the odors and stuff. And I thought that was a cool
Yeah, this one,
paper.
the interest in this one just got revamped because of uh Jeremy Plum and 80 Ray's paper. People are getting really interested in this smells, and that it's not just the terpenes, right? It's not all about the terpenes, it's about other stuff. And these figures are not as exciting as the ones that are in my dissertation. This is not how I would have presented this information, but Avery Gilbert, who is one of the authors on the paper, this is sort of his wheelhouse is the odor stuff. I'm just the geneticist on this paper. So I kind of let him take control of that. But I don't know if you have access to my dissertation, but those are much, much better. But so Avery approached me after my first paper came out, the one that shows that, within strain variation. And as soon as he said he was a sense scientist that specializes in aroma, I was like, oh, I totally know what you want to do. Like, let's go hunt down samples that are, we know are genetically the same, and then one that is genetically different. So Blue Dream, for example, get 5 Blue Dreams that are all exactly the same genetically, but find that outlier, and let's see if people can pick up on that difference. And either way, the story would be really cool. If they can pick it up and it does smell different, that's great. But if they can't pick it up and it smells the same as these ones that are genetically similar, maybe that's why, these things get called something that they're not, is because they're so similar to, something else. So that's the study we did. Let's see. Yeah. Go forward. Keep going, keep going. Keep going. Oh, yeah, go back. You were, keep going. Go like past halfway. More, 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 more. Keep going. Keep going. Keep going. All right. So this is the smell study. And this is something that he published before we did our study together, and he found, he was looking at putting cannabis into different categories. He came up with 2 scent categories, and then we did our work together. So yeah, if you go to that one. So I drove around Colorado and I had to try to find things that were the same. So we kind of picked things from his study that we knew smelled different and that I knew would be on shelves, because I had to, I had to purchase all this stuff out of pocket, and I didn't want to purchase like an exorbitant amount of things because I didn't have that much money. And we wanted to make sure that we would get some genetic outliers and things that were genetically the same. So the things that made it into what we were collecting were Durban Poison, OG Kush, Sour Diesel, Blue Dream, and Mob Boss. And if you go to the next one, yeah, that one. So this is that same kind of, graph that we looked at before. So all the things that are genetically the same, you're going to have the same colors. The ones that are genetically different have a different color. So for OG Kush, this 1SN is different than the 2SN, 3SN, 4SN, 5SN. So we were able to pick some OG Kushes that were the same and then include this outlier. Same thing for Blue Dream. Durban Poison, we actually used the 1, 4, and 5 because they were genetically spot on, identical, no letter differences, nothing. And we use that as a control to see how much variation is within things that are exactly the same. And then Mob Boss, we use that outlier, and then Sour Diesel, we didn't find an outlier, so that got tossed from the study. But because these were all from different dispensaries all over Colorado, we can assume that these are also from different cultivators, right? Because a lot of dispensaries have either their own cultivation or they get it from a nearby cultivator. On the OG Kush, this is interesting because the ones with the asterisks, they all had the same lot number on the package. So I think they were all from the same grower, potentially even the same grow. And yet this one was completely different, which is really interesting. Who knows what's going on there? Obviously, the metric isn't working.
Well,
Or
someone got their moms mixed up.
I think so. Yeah, something. So so that's what we did. And then so the smell part of the study was next. So we picked our samples that we were going to use. So then I took what we were going to use and then I randomized them with numbers and gave them to Avery. So he didn't even know what he had in jars. He just knew we got jars of weed. And then he found 55 people, a pretty good mix of males and females, all different ages, but they had to be under 50 and over 21. No smoking, no touching, just smelling. And they had like an iPad and it was pick all that you can smell. And people's sensory perception kind of peters out after about 15 smells. So we had to keep the sample size small so that people could be accurate in what the aromas were that they were picking up. And so these are the top pooled odor descriptors. And you can see there's more than 5, but that's because there's more than one sample. So it's the top 5 pool descriptors for sample 1, sample 2, sample 3. And so for German poison, these are the top 5 odor descriptors. And you can see there's a ton of variation. You know, obviously there's more than 5 odor descriptors, but even within these odors, like lemon, all over the place, tea, they're all over the place. And these are the ones that are identical, right? And Blue Dream, same thing, Mob Boss, all over the place, OG Kush, all over the place. So, we have a lot of variation in the smells, and it's not just limited to those outliers, but it is, the outliers do, so on this, all of these are genetic consensus samples, so the outliers not included. If we go to the next slide, if we go to the next slide, so this is the consensus mean versus the outlier. So you can see that the Blue Dream was like a lot, the outlier was like a lot more pungent and chemical and skunk than the consensus samples, the mean of the consensus samples. Mob Boss was a lot more sweet and berry and flowery for the outlier, and a lot less chemical and woody. And OG Kush, there were some differences as well. So you can see when you include the outlier, yeah, people can smell something different in those genetic outliers. So those genetic outliers are not just genetically different, they're also chemotypically different somehow as well.
It's really interesting with that, you know, they were able to pick that out.
Yeah, and then so I thought, well, that's a lot to take in, and so I wanted to kind of like distill it down a little bit more. And so I wanted to like group the smells. So these are what the groups that I came up with, and you can see that like the sweet category has a lot more in it than the other 4 categories. But what came out of this was really interesting. So if you go to the next slide. All right, so that's just describing what each is. So yeah, you can see like the outlier for Blue Dream was a lot more pungent. The other categories were pretty spot on. Mob Boss was less pungent but more sweet and floral, and OG Kush was less sweet but more floral and earthy. But I thought after that, like, I'm not a sensory scientist and I don't really know if my groups were good. So then I looked online for cannabis information, and I think it's Western Cultured has a wheel of terpenes, and most of these are on that terpene wheel. So I used their categories to try and categorize, and they're much more even in terms of what's in those categories. And same thing, the outlier was different. Those are manly scents, those bitter smells. So same thing. So Blue Dream is more bitter and less sweet. Mob Boss is more sweet, less spicy and bitter. And then OG Kush was pretty similar in most categories, but less bitter was the outlier. Less of those manly smells. And then what else did I do? Oh, I looked at the actual like cannabinoids and terpenes because I was like, this is great, but it's sensory analysis, which is kind of, it could be considered like woo-woo science. Like, how do we know? People are, you know, whatever. And so I actually looked at the terpenes next. The cannabinoids first, which you can see they're all over the place. So that was interesting. But I thought, well, maybe, you know, we don't know if they're harvesting them at the same time. We don't know how they're curing them. Like, we don't know any of that stuff. So, but it's good to know that, you know, you can get different cannabinoid profiles when you grow in different conditions. And then the terpenes, let's see, keep going a little bit. A little bit more, a little bit more. So we measured, I think it was 9 cannabinoids, and I think we measured 23 terpenes. Yeah, there you go. That's getting into the terpene stuff. All right, so these are just the terpenes. And I kind of wanted you to show like the variation among different strains. So like Blue Dream is in green, Durban Poison's orange, Ma Boss. And so those are just the averages, but that doesn't really show you the variation within strains. So if you go to the next little bit, you can see, okay, so this is Durban Poison. Again, were the three that were genetically identical, and you can see like terpinolene and beta caryophyllene and bisabol. I can never say that. There's a lot of variation in the terpenes. Again, different growing conditions. Terpenes are volatile, so how they cured them, how they stored them, the time to sale are all going to play a part in what terpenes are present. And so, like, this is a perfect time for me to say, you know, when you get a COA and it's got your terpene profile on it, that's not what you're taking home, and that's not what you're smoking. Because when they do a COA, it's generally, you know, right at the end of the grow. It's, you know, freshly dried, cured, whatever. It's going to sit on the shelf for a while, hopefully not too long, but you never know. Who knows how long they were, it depends on the state. Some states you have to send in the cured dried product in the packaging that you're going to sell it in. Some states, you know, you just do whatever. But the thing is, like, when you get it home, where do you store it? Do you store it in the fridge? Do you store it in a cool, dark place? Do you put it in a glass jar on your windowsill? Because that is going to completely change your terpene profile. So it's not like completely surprising that these are all over the place, but people seem to think that terpene profiles are you know, going to tell you something about this product, but it's not really giving you the information that you need at the point of consumption. Not to mention you're lighting it on fire and half of this stuff is going up in smoke anyway.
Oh yeah, plus you know how you grow it too is going to change the chemical profile.
Mm-hmm. Yep. Yeah, so the whole crux of like and then at the very, very end of this, well, like a little bit further on, you know, I was looking at the packaging when I did the cannabinoid content, I was looking at the packaging and it was like my testing says, you know, 14%, 10%, 15%. I was like, I don't remember buying anything with numbers that low. And so I compared the packaging to my test results and I was like, holy shit, like these are really different. And so from this, my my boss, my mentor was like, we need to expand this study. Like this could be anomalous. There's only 15 samples, but we need to expand this. You've got all hybrids here, first of all. He's like, we need to go get some sativas and some indicas. Go and get the lowest you can find, go and get the highest you can find. And let's try to figure out, is this, you know, is this an issue? So that study I just was able to publish about a year ago now. It's, you know, THC inflation, which it's been a big point of conversation the last few months or so. And finally, policymakers and regulators are sitting up and listening. Like, it's crazy. On average, the potency was 30% higher than what was actually in the product. And in some cases, 50% higher, which is, you know, like I mean, it's whatever, but, you know, people are purchasing things based on THC content. So if it doesn't have that much THC in it, it's, you know, maybe you're getting ripped off, but maybe you're not. But the other thing is like a lot of people buy flower and then go home and make their own medicine from it, tinctures or edibles. How are they supposed to dose their medicine correctly if they're not given accurate numbers? And yeah, it's flower and there should be a range that's reported, you know, but some of these ranges, like 15 to 25%, 15 to 26%, like 14 to 25%. Is that helpful for somebody? Not really. Like, that's a huge difference. But if you're actually taking buds from the bottom, a representative sample of all of the plants in your grow, and you've got 26% of the, apical flower versus 14% in the bottom branches, yeah, I mean, I guess you're gonna get that kind of range, but I don't think it's that. I don't know. I don't know. This is just kind of a call to consumers, buyer beware. This is something you should be aware of, kind of thing.
Well,
Um,
that too, and are any of the, what's the calibration, for the most of the people in the industry, right? I'm sure half of these machines aren't calibrated, right? It's not
well, you can
profitable for them
calibrate
to
it.
calibrate it.
I mean, there's no like standard protocol that everybody is supposed to follow is thing number one. There's differences in, instruments, there's differences in coils, there's differences in sample prep. There's all kinds of differences that could be reeled in, and you should follow these, protocols and methods, but there's all kinds of things where you can tweak it just a little bit. How much you dry the flower, the more you dry it, the higher your THC is going to be. If you're going to dry it down to sawdust, you'll get a higher number. Like our state tests for our hemp flower, the moisture content is anywhere from 5% to 11%. And I'm like, dude, like, come on, like pick one and so when we get our THC, and it's nudging up towards 0.3%, but it's at 6% moisture, I'm like, come on, like but anyway, yeah, so there's all kinds of places where numbers can be inflated from, little things here and there, calibration differences in instrumentation, differences in sample prep. But then there's some outright like fraudulent activity going on all the way up to just changing numbers on the COA. Uh, it first, consumers need to be aware of it. Two, regulators need to be aware of it. I don't know how it can be fixed, but there's a few suggestions. People are coming up with pretty good suggestions. So, one of the suggestions is to start taxing based on THC. So we're already, selling and purchasing based on THC. Why not slap a tax on there? Oh, okay, so you got 25% THC, you say that's going to cost you this much in tax. So then we'll see the swing the other way. With THC, and that's happening in California right now, as the THC percentages are going down because they're putting some other checks and balances in place.
Really interesting. I hadn't heard of that. That's definitely different. Um, you also did a lot of work with, or you're doing work with aquaponic and cultivation. Do you want to talk a little bit about that, or at least as much as you're
Yeah,
wanting to?
so I'm like I said, I don't know if I said it or not, but I'm technically not a cultivator. I do love growing plants. I like, you see my bananas back here. I'm not very good at it. I'm not good at growing anything. I'm really surprised my bananas are still alive through the winter. I'm just trying to keep them alive through the winter so I can put them back outside for the summer. Um, but I do work for a cultivation company, and they started out growing vegetables, so lettuce and tomatoes and peppers and things like that. And decided that they would switch and give hemp a run with the aquaponic setup, which has been challenging. It was a closed loop system. We've since decoupled the system, but the fishes are the sole source of nutrients for our plants. We don't do any, well, that's not true. We have brewed some compost teas and we've got, living soil. We're doing no-till method now. So we've got microbes, we've got worms that we have to feed to. So we do feed the soil. But in terms of feeding the plants, that's the fish's job. I test the water. So, and just keeping, the fishes happy with the pH and keeping the plants happy with the pH. There's a lot of nuances to keeping everything at the right levels and making sure the plants get enough nutrients and things like that. And then we have five houses, it's light depth in the forest. So there's always fun things that happen. Like one of our grows, we got a couple of mice came in and we had just planted and they came in and ripped up all the seedlings, chewed off all the tops, like a whole house was just annihilated in one night. And we were like, fuck. But they were small plants, so that put us what, 3-4 weeks behind, I think. Not cool, not cool mice. And then we've just had some slugs, which are completely under control. But I didn't know. I was like, well, where the hell did these come from? Because, they just popped up out of nowhere. I didn't realize slugs could do the asexual thing. I was like, okay, so all it took was one, and now we've got more. So it's been slug hunting. Every morning, slug hunting. I think one day we pulled out about 75, but most days it's anywhere between 2 and 5 that we find. So I think it's under control now, and it's just really concentrated in one corner. It hasn't spread. So that's good. But there's always something fun that goes on at the farm. Um, and yeah, we've just been kind of playing with different strains and different, just tweaking here and there methods and figuring out, the harvest window. What does 15 extra days growing time do? Does it, and is it worth it? Like, when you crunch the numbers, how much more mass do you get? How much more, cannabis do you, or cannabinoids do you get? And if we had those 2 extra weeks in another grow, would that be more worth it? So it's just, we're sort of in the research phase still. Um, fucking around and finding out. Why not?
With the slugs, I know we had there's I know in the UK, I don't know if you can get it in the States, there's a commercially available slug nematode for slugs and snails. But we had another person come on the podcast. I'm trying to remember who it was, that talked about collecting local slugs, putting them on like a little in a bucket and making like a little raft of grass or other plants and collecting a bunch of slugs and basically having a little island of slugs in the middle of your bucket or Rubbermaid tub. And then basically eventually you're going to get one that is infected with your local nematodes that do it, right? So once you see them all of a sudden have the population collapse in there of your little prisoner camp of slugs, that now you have the nematodes, then you can take that, blend it, and then apply it to the roots. And they had really good luck with that at a larger scale organically. I believe it was the gentleman from Vietnam, but it might be wrong. I'll have to remember
Do
and
you
go back and
remember
look
what
at my notes.
do you remember what the nematode? Because we have a blend of 3 species, I think, in our
Yep,
bed.
the triple strike. Yep. So the triple strike is more insect geared, but there's a bunch of different ones, depending on where you are in the world. But I don't know if there's one approved for in the United States. There might be. I know there was one that they were trying to get through in Canada. The one in the UK is, unless you have a friend that lives in the UK, and then maybe you can find other means, but you can legally go online and ship it to the States. So it is something to look into, but you can culture it yourself, which
Yeah,
is always a little more work, but also a little more fun.
we were so like I said, the population's under control at the moment, and they don't really seem to like the cannabis. Like they're not, I mean, they are eating it but there's nothing else to eat in there, right? They're in there and they're stuck. Um, Sometimes we'll get a mushroom or two and they'll go after that, but they don't seem super happy living off the cannabis. So we were gonna try if it was still out of control, we were gonna do the beer thing to see if that works, because I heard that works, and that we were just gonna bury like solo cups with beer and just you know, party with your friends and then die drunk. But we haven't needed to do that yet. But that's good to know about the nematodes, and that sounds like it could be fun to mess with.
That is something else too. I've noticed a lot when you're doing living soil with aquaponics is that you get lots and lots of mushroom blooms because of the humidity being higher and the moisture content kind of being a little bit higher. You get just tons and tons of blooms of different ones. Maybe you want to touch on that. I always found too that when I have those blooms going off, And the pots, we very rarely have botrytis issues or powdery mildew issues or anything like that on those because you're getting that extra stimulation from the fungi and things like that. It's always fun to kind of see how that affects the garden as a whole.
Yeah, so I was actually laughing because some of the pictures that you showed of your upcoming class, you've got the little mushrooms coming out of the pot. I just took pictures of something like that the other day. We were like, oh wow, that's so cool. But yeah, we do get different kinds of mushrooms. I just finished a guide to pest pathogens and problems because our employees were like, how do we know like what we're looking for? And when we see something, how do we know if it's good or bad? Like what's a good because I just showed them a baby ladybug and our farm manager was like, holy shit, I would have killed that. I would have had no idea that was a baby ladybug because they look really like little monsters. They don't look like ladybugs at all. Yeah, we've seen those. Um, so I made a guidebook of, nutrient issues, mushrooms, good bugs, bad bugs, mildew, mold, um, what other stuff do I, what rodent damage looks like since we had not that we could have done anything like they were in there one night and it was just two of them and we caught them right away. And we have thumpers. I didn't even know those were a thing. Do you know what those are?
No, what are those?
So they're
Something
these
else to be afraid of.
no, it's a way to keep the mice away. So it's this thing that you bury in the ground, and I think it's every 30 seconds or so, it vibrates and it gives off this like high-pitched squeal, and it scares the rodents away, and they don't come near the farm because it shakes the ground and it squeals, and they think it's something horrifying. So we have them around the perimeter and in the greenhouses, and it just goes like every 30 seconds or so. And it's just a way to keep everybody away.
That's interesting. Yeah, I guess it attracts the sandworms.
Well, I don't know. I don't think so. I don't know.
I think this is one of the better ones I've taken.
That's a nice one. Yeah, that's kind of what yeah. And I don't know, like I know that mushrooms are a good indicator of soil health, but I'm never sure should I pull them or should I leave them?
I've always just left them, unless they're really large and gonna rot or something on part of the plant. Otherwise, I usually just let them spore out and do their thing. I've never seen any kind of negative impact. I'm sure there is a fruiting body mushroom out there that'll screw up your plants, but I haven't run into it yet.
Yeah, uh, I mean, I guess we're of the thought that if we don't know, we pull it, you know.
Hmm. Yeah,
But
that's
I like
interesting.
them. I like finding them and I always photograph them before I pull them.
I definitely looking forward to seeing your work one day in Jersey. You live not too far from my family, so I'll have
Yeah,
to pop by one
you
day.
said you were going to come visit, and the weather's going to start getting nice here pretty soon. So you have to keep your word, and I know my boss is excited to meet you too.
Oh yeah, I'm
Yeah.
definitely would love to do that and see your the hard work of your team there.
Yeah, I mean, we have come a long way from just a year ago. I don't know how many greenhouses we had up and running last time I talked to you, but when I got here, it would be 3 years ago, we had one greenhouse that had vegetables in it, and then the other greenhouse had been built for hemp, but wasn't really up and running. It had like two small raft tables, and that was it. Um, since then, we've built three like greenhouses, twice, at least twice this, more than twice the size, three times the size probably of those smaller greenhouses. And they probably put those up. Oh God. I mean, I don't even know if they were up and built just a year ago. We might have had one of them. But yeah, they put them up like and it's really just like our farm manager and Mike that built everything, just the two of them. We didn't have, a construction crew and I certainly didn't help because can't lift heavy things and I'm almost 50. But no, I mean they kicked ass and they're up and running and we've got beds going and all the greenhouses. I think we're on our, I don't know, third or fourth or fifth full grow in greenhouse one, I think. So it's pretty fun, and changing over to living soil, I think was probably one of the best things we did. I really enjoy that element of having a whole, ecosystem that should take care of itself if you take care of it, kind of, instead of having to add all kinds of crap just to get your plants to grow.
Oh yeah, I definitely understand. I know most of the facilities I'm working with, we're getting, between 80 and 90% of the nutrients from just the fish alone. So I definitely feel you on that. Definitely make things look complicated once you have it figured out.
Yeah, yeah. I mean, we've been having a problem with our fish, but we've been having problems with our pH as well. With all of the rain that we've had, it's diluted our well water, which is usually a higher pH. So when we fill up, it raises our pH. And so it's been kind of a balancing act with all of that, and our fish have not been super happy. Then we had like some cold weather that, the water got cold, so then the fish get lethargic, and I mean, it's always just, things, life.
I definitely remember dealing with all that when I did aquarium maintenance there in the Philadelphia area, dealing with those same water table issues and all the rest that you're describing.
Yeah, yeah. It's but we sample the water every day and do what we can to try and keep it as stable as we can. So, yeah.
What type of fish are you guys running?
We have a mixture of koi and tilapia. Uh, I think that's it. Maybe we have a third type in there. I'm not sure. But yeah, it's just a hodgepodge mishmash of all these pretty little dudes that swim around and fight with each other.
Very cool. Well, I don't want to keep you up too late. I know you're there on the East Coast, so and it's getting a little bit late there. So, is there anything else that you wanted to mention or have coming up before we wrap up the show? I know you're regularly on the Dank Hour. Do you want to mention that as well?
Yeah, so we have a show every Tuesday night called the Dank Hour, with it's with London Nero, who is in Croatia. He's our host. And then every week we have some, a guest of some sort. So we have London, Johnny, Mark Scaldoni, who's a chemist, Evian Ida, cultivator, Kyle Boyer. And Tess is no longer with us, but she's with us in spirit. She's not dead or anything. She's just doing other things. But yeah, we have a guest every week. We're actually taking a 6-week hiatus because London needs a vacation, but we'll be coming back with new guests after our little break. But all of these are available on YouTube. There's some really good guests. We've had, let's see, we've had Ethan Russo on the show, um, Ed Rosenthal, uh, Reggie Gaudino, Jeremy Warren right there, Connor Stevens, who's a tissue culture expert. The Trusted Canon Nurses came on. Yeah, we've had some really great guests. So those are really fun to watch. And then I've got stuff on my website, various, including our chitty chats, interviews, my dissertation, my publications, my defense of my dissertation, which is what you were running through that video. If you want to watch me defend all of my research at the end of my PhD, I've got, yeah, some of my botanical art is up there. So that one's pretty cool. The herbarium specimen, that's all a drawing. It's like a whole like 3x4 sheet of paper, and it's, if you go back one, so that's a flat piece of paper. That's just one flat piece of paper, and everything on there is drawn on, except for the cork board that's watercolor. But that's all flat. So that's like one of the coolest things I ever did. So all that's on my website, which is just annaschwabe.com. And you can find all kinds of links there and stuff and find out what I do. So like I said, I'm from Australia. So that piece that you were just looking at, I had an American side and an Australian side. And then there's another layer that goes on top of that. And it's like these bush babies. There's an artist called May Gibbs, and she does these little like bush babies, they're little flower children. And I did this piece that where they're like talking to each other. But so yeah, there's the Australian side is with the budgie and the little possum, and then the American side is with the butter and eggs and the little squirrel and the little hummingbird. Yeah, cute. The West Australian pygmy possum. So, and I've only done one cannabis work. Uh, and that was for a friend of mine. He took the picture and I drew the picture. He took like the photograph and I drew the picture from it. But that's pretty cool.
Oh yeah. And you guys can find that at annaschwabe.com. Listen to this in audio format.
Yeah.
Huh. Awesome.
Yeah.
Thank you so much for coming on the show this evening. I really enjoyed speaking with you again. Always good to have you on the show, and
Thanks.
definitely excited to come check out your work.
Thank you for having me on again. I always, I talk a lot, but I get so excited talking about it. I think I could talk for hours, probably just like you can about cannabis. And um, yeah, thanks for having me on. I really, truly appreciate it. And I can't wait to actually meet you in person one day in Boise.
Yeah, it'll be a lot of fun. I'll have to go get a cheesesteak or something then.
I'll have like a pizza. All right.
Well, thank you so much. And you guys can also find her at anna.thecannabis.geneticist on Instagram as well,
Hmm.
if you want to follow her work.
And I'm on
So,
LinkedIn as well. Yep, yep.
awesome. Well, thank you so much. I really appreciate it.
Thank you.
All right. And one last time as well, be sure to check out our Aquaponic Masterclass May 22nd through 26th in Beggs, Oklahoma at Olive Branch Aquaponic Facility. I'll be teaching a long format first 5-day class that we've ever taught. It'll be a lot of fun. They also work with University of Oklahoma. So this is a accredited um, farm that we're teaching with. They work with other universities. They also work with Langston University. So they do a lot of neat stuff. And I've known Bob for quite a few years, and he's a great guy. And it's going to be a lot of fun to teach with him. And I'm really looking forward to the course. If you're looking for more information on top to bottom on a wide range of different crops, the biggest that we've ever taught before, you can check it out here at aquaponicmasterclass.com. All right, everybody. Thanks a lot for watching. We'll be back again next week. Yeah, I hope you have a great week. Again, you can find the show on Ponics and all the things. I am working on getting the rest of the episodes up on Rumble and Odyssey. So, check those out as well. If you don't want to watch it on YouTube on the video format.